Hey everyone, let's talk about enzymes. They're like tiny, super efficient helpers in your body, speeding up chemical reactions that are essential for life. Think of them as biological catalysts, primarily proteins, that make reactions happen much faster. They do this by lowering what we call activation energy, the energy needed to start a reaction. Importantly, enzymes don't change the end result or stability of the products, they just get things moving quicker. Key takeaways: enzymes are protein catalysts. They speed up reactions by lowering activation energy, and they don't change the final products. So, how do enzymes know exactly what molecules to work on? It's all about specificity. Each enzyme has a special area called the active site, which is shaped perfectly for a specific molecule known as the substrate. This ensures the enzyme only acts on the right molecule, preventing it from working on the wrong ones. We explain this with two models. First, the lock and key model, where the substrate fits perfectly into the enzyme's active site, like a key into a lock. The active site is rigid and shaped exactly for the substrate. The second model is the induced fit model. Here, the enzyme's active site isn't completely rigid. When the substrate approaches, the enzyme actually changes its shape slightly to better fit the substrate, forming a tight complex. This is more flexible and accurate for many enzymes. Let's see how this works. The substrate approaches the enzyme's active site. In the induced fit model, the enzyme adjusts its shape to fit the substrate perfectly, forming an enzyme-substrate complex. This precise fit ensures the reaction can happen efficiently. Once bound, the enzyme-substrate complex forms. The enzyme helps the reaction happen, and then the products are released. The enzyme is unchanged and ready to work again. This specificity is crucial. It means enzymes don't waste time on wrong molecules. Key takeaways: enzymes have active sites shaped for specific substrates. The lock and key model shows a rigid fit, while the induced fit model shows the enzyme adapting its shape. This specificity ensures enzymes work efficiently on the right molecules, like a key opening only its matching lock. Sometimes enzymes need a little extra help to do their job. That's where cofactors and coenzymes come in. These helpers bind to the enzyme and assist in the reaction, making the enzyme more effective. Cofactors are usually inorganic, like metal ions. For example, magnesium ions help ATPases work properly. Coenzymes are organic and often derived from vitamins. NAD made from niacin helps oxidoreductases in redox reactions. Key takeaways: cofactors are inorganic helpers like metal ions, coenzymes are organic helpers from vitamins, and both assist enzymes in performing their jobs effectively. Enzymes are organized into 6 main classes based on the types of reactions they catalyze. Let's explore each one with some examples to see how diverse enzyme functions can be. First, oxidoreductases catalyze redox reactions, where electrons are transferred between molecules. This is crucial for energy production in cells. Here's an example showing electron transfer. Next, transferases move functional groups from one molecule to another. A great example is how hexokinase transfers a phosphate group from ATP to glucose, using magnesium as a cofactor. Third, hydrolases break down molecules using water. This includes enzymes that digest food or break down sugars, like in this glycosylation reaction, where a sugar is cleaved. Fourth, isomerases rearrange molecules without changing their atoms. For instance, alanine racemase converts L-alanine to D-alanine, important for bacterial cell walls. Another example splits fructose into 2 phosphates. Fifth, lyases cleave bonds without using water, often creating double bonds. These help in breaking down complex molecules in metabolism. Finally, ligases form new bonds using energy, often from ATP. They join molecules together, crucial for DNA replication and repair. To wrap up, these 6 enzyme classes, oxidoreductases, transferases, hydrolases, isomerases, lyases, and ligases, cover the full range of biochemical reactions. Knowing them helps us appreciate how enzymes power everything from digestion to DNA repair in our bodies. Enzymes are sensitive to temperature. Most human enzymes function best around 37 degrees Celsius, which matches our body temperature. This is their optimal temperature where activity peaks. If temperatures get too high, above about 40 degrees Celsius, the enzyme structure changes. This denaturation means the active site no longer fits the substrate, and the enzyme stops working. pH levels are equally important. Most enzymes have an optimal pH range, typically neutral between 6 and 8. Extreme pH can also cause denaturation, just like extreme temperatures. Different enzymes have different optima. For example, pepsin in your stomach works best at very acidic pH around 1 to 2, while trypsin in your intestines prefers neutral pH around 7 to 8. To wrap up, enzymes perform best at their optimal temperature and pH. Go beyond these ranges and denaturation can make them useless. Remember, different enzymes have different optima depending on where they work in the body. Enzymes are powerful catalysts, but their activity needs to be carefully regulated to maintain balance in the body. Let's explore three main ways this happens: inhibition, allosteric control, and zymogen activation. First, there's inhibition, where molecules bind to the enzyme and reduce its activity. In competitive inhibition, molecules mimic the substrate and compete for the active site. For example, statins block the enzyme HMG-CoA reductase, which is important for cholesterol production. In noncompetitive inhibition, inhibitors bind to a different site on the enzyme, changing its shape and preventing catalysis. An example is cyanide, which blocks cytochrome oxidase in the electron transport chain. Second, allosteric control involves molecules binding to a site away from the active site, causing the enzyme to change shape and either increase or decrease activity. For instance, ATP can inhibit phosphofructokinase, an important enzyme in glycolysis, helping regulate energy levels. Finally, zymogen activation means enzymes are produced in an inactive form to prevent premature activity. They're activated only when needed. A classic example is trypsinogen, which becomes the active enzyme trypsin in the digestive system. To wrap up, enzyme regulation ensures proper timing and balance. Competitive inhibitors compete for active sites, non-competitive ones bind elsewhere. Allosteric effectors change enzyme shape, and zymogens provide safe activation. These mechanisms keep our bodies functioning smoothly. Let's look at some real-world examples of enzymes in action. First, Taq polymerase is a heat-stable DNA polymerase enzyme found in Thermus aquaticus bacteria. This enzyme is crucial in PCR, or polymerase chain reaction, which amplifies DNA for testing and research applications. Next, Cas9 nuclease comes from the CRISPR system and acts like molecular scissors, cleaving DNA at specific locations for precise genome editing. This enzyme revolutionizes gene therapy, treating genetic diseases and improving crops. PETase and PETase are enzymes from Ideonella sakaiensis bacteria, discovered in 2016. They work together to degrade PET plastics, breaking down plastic waste into smaller, reusable components for recycling. Finally, immobilized lipases are enzymes attached to beads for repeated use in biofuel production. In a 2026 case study from Brazil's biofuel industry, this approach achieved a 95% yield, significantly cutting energy costs compared to traditional chemical catalysts. These examples show how critical enzymes are in medicine, biotechnology, environmental solutions, and industry. From amplifying DNA to recycling plastics and producing biofuels, enzymes solve real-world problems efficiently. The world of enzymes is constantly evolving. Current trends include enzyme immobilization for reusability in industrial processes, directed evolution to create specialized enzymes, and AI enzyme hybrids that combine artificial intelligence with enzyme design. Future applications are exciting. Enzyme-based biosensors will revolutionize diagnostics, detecting diseases with incredible sensitivity. Engineered nitrogenase enzymes could transform agriculture by reducing the need for synthetic fertilizers. And enzymes are key to green chemistry, like cellulases, producing biofuels from plant waste, cutting our reliance on fossil fuels. Key takeaways: enzymes help reduce fossil fuel dependence through sustainable processes. AI-driven enzyme design addresses global challenges like hunger and pollution. This field is incredibly exciting to follow. As biochemist Jennifer Doudna said in her 2025 TED Talk, enzymes are the future of sustainable technology. Designing them with AI could solve global challenges like hunger and pollution. Keep an eye on this exciting field.